All right, so we're going to explain DNA synthesis. Just going to get straight into it. So DNA synthesis can be explained in 3 easy steps. So we'll get started. So the first one I'm just drawing here is going to represent your double-stranded DNA. Looks like a ladder. Essentially what's going to happen is you're going to have an enzyme, helicase, and this enzyme is going to be responsible for unzipping the DNA. It's going to break the hydrogen bonds that are joining the bases together. So once that happens, you have step 2, where you have now the double-stranded DNA, which is now separated. Easy? Okay, so because this is all happening in the nucleus, you know that you're going to be having the bases freely roaming around in the nucleus. So I'm just going to draw those at the bottom here. So just like A, T, G, C, do the same 4 again, because there are only 4 for DNA. Okay, so they're unzipped. You're still going to have the bases 1 on each side. So let's just say this is A, this is G, T, C, and let's just say, for the sake of it, because they are complementary, they are opposite. So what's going to happen is these bases that are freely roaming around in the nucleus, remember, they are complementary, so they are just going to match up. So for example, with this A, this base that's floating around is going to match up with it there. And for this one, it'll be like that. This will match up here, and G is going to match up there. And then same thing on the other side. So T is going to match up with A's, G is going to match up with C, T again with A, and C again with G. Hopefully you can sort of see that, but it's the complementary base pairing. So that's your key for step 2, complementary base pairing with your bases that are freely roaming around in the nucleus. Now, step 3, essentially, because I've used different colors here, what you have now is I've drawn it in red. Just like here, because the bases here in step 2 were in red. So essentially what you have now is you have you have identical copies of the DNA, and the reason that this is known as semi-conservative, if you take a look based on the colors, you've got one part of the original strand, which is in purple. So this is why it's called semi-conservative, because you have a bit of the original strand of DNA that you had in step 1, and then the red bits are the bases that were complementary and matched in the second step. So that's basically it. 3 easy steps for DNA synthesis. 1, helicase unzips it, splits up the DNA double helix structure. Step 2, you've got the free bases floating around in the nucleus. They're going to match up, complementary base pairing. Step 3, you now have the 2 new strands. There also is, I almost forgot, an enzyme involved with this, and it's known as DNA polymerase. And DNA polymerase is responsible for making sure that the strands end up joining back together. So like we said, like helicase was responsible for separating the double helix, DNA polymerase is responsible for joining the 2 strands back together again. Okay? So hopefully that's it. Step 1, helicase, breaking hydrogen bonds. Easy way to remember it, helicase, starts with an H, hydrogen bonds. Okay? Second step, complementary base pairing with free bases in the nucleus. Step 3, enzyme DNA polymerase helps to join the 2 strands together, and it's called semi-conservative because you have part of the original strand in the 2 new copies in step 1.